Okay, so here we go. Why did I ask these questions? Oh no, hold on here. Bluetooth, I tell you. Okay, so dumb humans, frog DNA, fossilized dinosaur DNA, dumb humans, rage virus, chimpanzees, dumb humans, smart computers, weapon defense system. Here we go, number 1. Most of you got that one right, okay? Jurassic Park. Number 2. Nope, number 2 is down here. I'm the product of a human-made virus. Do you smell blood? It's 28 days later. Okay, so they do. It's kind of a scary movie. The third one was Terminator or any one of those. I'm a newfangled robot. Hasta la vista, baby. So what do all 3 of these have in common? Things get out of control. Okay? They're like, hey, this is going to be a nice amusement park. We can make money off this. The animals will keep them contained. Nothing could happen, right? Wrong. Okay, disgruntled employee sabotages it. Mayhem breaks loose. Down here, I don't know why they were studying a human-made virus and exposed it to chimpanzees that escaped. Gets out. Yeah. Terminator, again, allowing computer systems to have access to everything and to think on their own, right? So this section is regulating the cell cycle. I'm going to talk about how that cell cycle is regulated and what happens when it's not regulated, aka cancer. Okay, so our early observations of how cell cycle was controlled was that we started to look at cells underneath a microscope. And we noticed that when they grow and divide, they will stop those processes when they come in contact with another cell. It's kind of like, oh, sorry, that's your space. Okay, didn't mean to. Okay, cool, that's your space, here's my space. And they coexist very nicely with each other. During injury, okay, cells along that injury gets stimulated in some way and start to divide rapidly to repair those damaged cells, and they do this until the healing is complete. So those are kind of like naked eye observations. But what's really going on here is that we have some regulatory proteins that are being released. So there are lots of different proteins that do this. So dozens of proteins that tell the cell when to divide, and they are found inside and outside of the cell. Those that are found inside are called internal regulators. They respond to events that happen inside the cell. They act as checkpoints. You know, when a cell cycle goes through its phases of interphase. G1, hey, am I large enough? Should I start copying my DNA? All right, move on. Hey, I just copied my DNA. Did I do it correctly? If so, move on. And so they have little checkpoints where the cell's, just checking itself to make sure that it's doing what it's supposed to do. It can't enter mitosis, that's what M means, until the chromosomes are replicated. So DNA replication has to occur in order for it to move on to the next phase. We also have external regulators that respond to events outside the cell. They control how fast or slow that cell needs to divide. So when you're going through growth and development, you hit that puberty stage, you have hormones that are in your brain that get released into the bloodstream, and then they travel to those cells. Hey, we're going to grow here, okay? So growth factors. Whoops, I cut that off. There we go. Growth factors important during embryonic development and healing. So growth factors are an example of an external regulator. So here you can see a cell that's making those hormones. It's traveling through the bloodstream. It hits the target cell, and that target cell will respond to that. Okay, another kind of protein that is responsible for the cell cycle are called cyclins. We have noticed that cyclin levels change as a cell goes through the cell cycle. So levels are relatively low in G1, but then somewhere in the middle of it, it starts to increase rapidly. And it increases until the middle of S, and then it drops off the map. It stays low until G1 again. So something about cyclins that help it target the process of synthesis. So I'm going to zoom in here. Okay, I don't know if you have to write anything. So when you don't have any cyclin, okay, actually I'm going to move up here. Okay, so this is cyclin. And what it does is it interacts with another molecule called kinase. So when they are together, it's called cyclin-dependent kinase. So in this diagram, when you don't have cyclin, CDK is inactive. And so S is turned off. When you do have cyclin bonded to CDK,
You understand?
now it targets cells to begin their S phase. Another way cells can regulate themselves is through apoptosis, programmed cell death. So let me just talk about, there are two ways that cells can die. Okay, the first one's off as an accident. It gets damaged, it gets injured. It's very, very messy when this happens. So think about like bruises or contusions. Those are cells that are damaged, and so their contents spill out. That's why bruises go through different shades of color, or cuts that can also be kind of messy with the scab over and all of that. The second way that a cell can die is it's programmed. Okay, it's programmed to die, and we do this during development. Okay, you are all born with webbed. Okay, not born, but in your development process, we all had webbed fingers at one point. Okay, and these cells that are in between your digits went through apoptosis, and it kind of helped us sculpt our body. Now, you may have heard of people that have webbed toes. Yeah. And that just means that those cells did not get the signal to go through apoptosis, and so they were born with webbed toes. Okay. Other examples: frogs, tadpole. These cells will go through apoptosis. The tadpole will lose its tail and develop into a frog. This is a microscope slide just showing how a cell is going through apoptosis. It's very, very clean. It starts to shrink, and then it blebs off. It packages itself into these nice little vesicles until the cell no longer exists. So I think I have a video here. Hopefully it works. I didn't check. Okay, I don't think there's sound. How come I can't see? Dismiss. Okay, so pretty soon you're going to start to see it collapse upon itself, and then it's going to bleb off. Yep, like right here. So they're just all packaging themselves into nice little vesicles that will get reabsorbed elsewhere or eaten up by a lysosome or something. So these are all cells that are going through programmed cell death. Okay, so when a cell doesn't respond to external regulators or internal or cyclins, then cancer results. So cancer is just uncontrolled cell growth. It's not responding to those signals. The cell goes through the cell cycle like literally pedal to the metal, doesn't even check itself, goes right through all those checkpoints. So as a result, you end up with cells that grow uncontrollably, and that is one of the reasons why tumors are a characteristic of cancer. Now there are two types of tumors, benign and malignant, and I'm sure you've heard these terms before. Benign means it's non-cancerous and it doesn't spread. Malignant means it is cancerous and it can invade and spread to surrounding healthy tissue. And even if it's close enough to a bloodstream, travel through the circulatory system to infect another area of the body. So what these tumors do is they absorb nutrients, they block nerve connections, they ignore the personal space of cells, and they basically pinch healthy cells to death. That is what cancer is. If you think about the zodiac sign for cancer, what is it? A crab, pinchers. And so cancer is called cancer because it literally pinches healthy cells to death. So up here, why did I talk about apoptosis before? Sometimes cells can recognize that they have unfixable damage, okay? And so if a normal cell says, you know what, I got a lot of damage, mutations occurred, this is not going to be good for the organism, it will go through apoptosis. But cancer cells arise when they ignore that unfixable damage, and they go through the cell cycle, and it just continues to divide and divide and divide. Here's a biopsy picture just showing benign tumors. You can see multiple cells kind of crowded together, but it's not cancerous. Over here with malignant, it's tapped into the bloodstream. It's darker in color in appearance. So here's just another diagram showing malignant. They grow locally. They don't spread by invasion or metastasis. Over here with malignant, they invade tissues. They enter bloodstreams, and they can break off, as you can see here, and travel to other parts of the body. So what are the causes of cancer? We have figured out that it has to do with a defect in multiple genes, not just one. But one really well-known gene that has the mutation is called the p53 gene. And this gene is responsible for regulating cell growth and division. It can also be brought on to exposure to carcinogens like smoking, radiation, viral infections. Viruses are probably responsible for at least 20% of cancer worldwide, as well as heredity. Genes, if you have a mutation in your gene and it happens in the gametes, the egg or the sperm, yeah, you can pass that on to your offspring, so they get the faulty gene as well. Okay, so I'm going to zoom in on this diagram here that talks about normal p53 versus the mutated form. Okay, so if there's DNA damage, okay, or any type of abnormality, p53 recognizes that and it will halt the cell, okay? And wherever it is in the cell cycle, it's going to stop it, it's going to fix that damage, and then it will restart the cell cycle. If for some reason the damage is just too bad, then it will go through apoptosis and the cell will go through programmed cell death. In a mutated p53 gene, now we have DNA damage or some type of abnormality, and it doesn't recognize it, and the cell cycle will continue, and it will continue to create cells that have that mutation or that damage. So you get a whole bunch of cells that just ignore signals from your body to stop. So the cells become cancerous. Treatments of cancer, 3 different ways you can kind of think of it. Localize, so they see the tumor, it's in one area, so they'll go in through surgery and try to remove all of the tumorous cells that belong to the tumor. Second treatment, targeted beams of radiation. And the third is chemotherapy. Now, chemotherapy, I'm sure you've seen, people that have gone through it. They lose their sense of taste, sometimes smell. Their hair, they tend to lose their hair. And what's going on here is with chemotherapy, they get an IV, and inside the IV is a chemical that targets rapidly dividing cells, but it targets all cells that divide. So it's not just cancerous. I mean, yes, it targets the cancer ones, but healthy cells die in the process. And so your hair that's constantly growing, well, now those cells are not dividing and the hair follicles, they fall out. Or taste buds, you shed a lot of tongue cells, when you eat, but now they're not replacing themselves. So eating's, it's just hard to eat things when you can't taste or don't have an appetite. Your stomach cells, they replace themselves every 3 days, right? Because of the acid in your stomach, and that's another reason why they just tend not to, it's really difficult for them to eat. So that's it for the notes. Go grab your books. I forgot to tell you to grab your books.